So once upon a time, all of us were one cell big. That one cell then underwent mitosis to make two cells. And these two cells then had the same number and kinds of chromosomes as that first cell, that zygote that we once were had. And then they divided again and again, and now here we are 100 trillion cells later. So obviously we depend on mitosis a great deal. But for sexual reproduction to occur, a second cell division type is required, that of meiosis. Meiosis is going to be different. There are going to be two rounds of cell division. There will be prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. And as a result, there will be 4 daughter cells, but they will be different from the first cell that started meiosis. They will only have half the normal number of chromosomes. In humans, the only cells which have half the normal number of chromosomes are sperm in males and ova in females. And so when 23 chromosomes from a sperm fuse with the 23 chromosomes of the ovum, so if here's an ovum and there are 23 chromosomes, and then the sperm fertilizes this ovum and adds its 23 chromosomes. So here are the 23 chromosomes from the ovum. If 23 chromosomes from the sperm are then added, then a new cell with 46 chromosomes results, which is diploid, with one of these, say chromosome 1's coming from the mother, one from the father, one of these chromosome 2's from the mother, from the father, etc. All right, so that's the quick overview. This is required because most body cells have those 2 copies of each chromosome, because that's what the zygote has, and every time the cell undergoes mitosis, then that's what they possess. If these individuals would like to have a child, they engage in a deep French kiss, and the cheek cells from one individual fuse in the mouth of the other. Do we make a baby? When I ask my students, they often think for a second and sometimes look at their lab partners in new ways. But the answer is no. And the answer is that while those cheek cells were great cheek cells, you couldn't stick them together and now make a zygote. The main reason being there would now be too many chromosomes, because each of them had 2 of each chromosome, 2 copies of chromosome 1, 2 copies of chromosome 7, chromosome 13, chromosome 21, etc. Nor could you take fibroblasts from connective tissue, nor could you take nerve cells. The same problem would exist. Most body cells have 2 copies of each chromosome, and while that makes them wonderful muscle cells, bone cells, brain cells, etc., they would make lousy sex cells, and the odds are that the embryo would fail. It is only cells which have half the normal number of chromosomes, sperm and ova, which could then fuse. So notice here, there's the same kinds of chromosomes. So here there are 4 different kinds. So that's 1 kind, 2 kinds, 3 kinds, 4 kinds. Here there are 4 kinds of chromosomes. Here there are 4 kinds of chromosomes. But these cells have 1 of each kind, as opposed to all of these others, which have 2 of each kind. Those are the cell types which are required for sexual reproduction. The terms we use for these cells are haploid and diploid. So diploid cells have 2 copies of each chromosome, while haploid cells have 1 copy of each chromosome. For humans, that means that the 23 kinds of chromosomes, that's what we find in humans, 23 kinds, that a haploid cell would just have 23 chromosomes, 1 of each kind. But when a sperm with its 23 chromosomes fuses with an ovum with its 23 chromosomes, now you get a total of 46. Now, not any old 46 chromosomes. There are 2 of the longest chromosome, which we call chromosome 1. 1 came from the sperm, 1 came from the ovum. There are 2 of the second largest chromosome, chromosome 2. 1 came from the sperm, 1 came from the ovum. So it's not just any old, it's not just 23 chromosomes. It's specifically chromosome 1 and chromosome 2 and chromosome 3 and chromosome 4, which all vary in the genes that they have. And so, to begin our discussion of meiosis, we have to distinguish between cells which are haploid. Haploid cells have one copy of each chromosome. Here's chromosome 1, chromosome 2, and chromosome 3. Notice they have different structures, different banding patterns. They would then also have different genes. And then diploid cells. Diploid cells have two of each chromosome. Our cheek cells, our bone cells, our brain cells, our muscle cells would be diploid, but our sex cells would be haploid. Mitosis starts off with a diploid cell and then produces two diploid daughter cells. So they are clones of the mother cell. They have the same number and kinds of chromosomes. In contrast, meiosis gives us different cells, cells which are haploid, cells which have one copy of each chromosome. Now, before the any cell division occurs, we need an S phase, which replicates the genetic material. So in mitosis, the reason for this is that if we didn't replicate the genetic material, we would soon run out of chromosomes and out of genes. So if we didn't have an S phase, then when that one single-celled zygote divided to make a two-cell embryo, then each cell would then have half the normal number of chromosomes. When we divide again, we'd have to split the chromosomes in half again. So now it would be a quarter of the normal number, etc. And so every time cells divide, they would end up with less and less DNA, fewer and fewer genes. And so there's no way that we could get to our adult selves where the brain cells have all 46 chromosomes and all human genes. The muscle cells have all have 46 chromosomes and all human genes, etc. So all of your diploid cells have these chromosomes. So we need an S phase before mitosis, and we also then need an S phase before meiosis. Leading up to meiosis, we go through a G1 and S phase. So now that each of these then has a partner. So if this is the chromosome 1 that someone has from their mother, in the S phase before meiosis starts, it copies itself. So now there's a pair of sister chromatids of chromosome 1 you got from your mother. There's a pair of sister chromatids from the chromosome 1 you got from your father, etc. Now, I'm making this video for my genetics class, and so I know that they've covered meiosis before, and I'm just going to go over it in a little more detail than what they've covered before. And so one of the main areas is out of all of the phases of meiosis. So now we have prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Prophase 1 has so many important things happening that it gets divided into subphases. So prophase in general is longer than metaphase, anaphase, and telophase combined in mitosis. All right, so prophase is already the longest phase. But prophase 1 of meiosis is critical, and so it requires a bit more discussion. So in the first phase of prophase 1, known as leptotene, we start off with the chromatin condensing to form chromosomes. But the chromosomes are still rather narrow at this point. They're not as condensed as they will be in the next phase. So DNA in interphase is unwound chromatin, and it condenses to form chromosomes. And at the very first part of this, and this these are cross-sections of seminiferous tubules where sperm are being produced. In leptotene, the chromosomes can look thread-like because they're still very thin. Lepto in Greek, I believe, means thin. The next phase is what's called zygatine, and something very important is happening here. Actually, this formation of the synaptonemal complex, it actually begins in leptotene and ends in pachytene. But zygatine is where, it's coming together. So let me discuss it here. So here's the thing. In mitosis, you have a pair of sister chromatids that, for chromosome 1 you got from your mother, and a pair of sister chromatids, chromosome 1 you got from your father. And when they line up the metaphase plate, one could line up here, one could line up here, or reverse, or over here. So they don't have to be together, but in meiosis, they line up like this, right? So that they will form a group. This pairs of sister chromatids that you got from either parent, they will actually stick to each other. And so there is this complex made of proteins. First, you have lateral elements, which will attach to the sister chromatid you got from your mother. Here's one of the two. Here's one of the two sister chromatids you got from your father. So it attaches them, and then we form these transverse elements which stick from them, and then there's a central element which now joins, so that by the end of zygatine, these are now joined, even though they were once separate. This is the pair of sister chromatids for chromosome 1 you got from your mother. Here's from one you got from your father. And in mitosis, they would stay separate, never associate with each other. But in meiosis, they are going to form a complex where they are physically joined for a bit, a synaptonemal complex. So meiosis is going to be distinct, not just in the number of cell divisions, 2 as opposed to 1 in mitosis, not just in the daughter cells being haploid as opposed to diploid in mitosis, but also because of a number of other differences which occur along the way, such as the formation of this synaptonemal complex, which will hold these pairs of sister chromatids together. In this video, I just name the separate elements and point out that they're important. Mutations in the genes for these elements can be a cause of infertility, can be a cause for gonads to fail and not develop, could be a cause of miscarriage. And so this is important. And I'll build on this later, but just, big point is this has to happen. So when we talk about, the homologous chromosomes pairing, the crossing over, that's kind of interesting. No, it has to happen. If it doesn't happen, this cell will probably die. It will not make gametes, but it will pause in the middle. And the cell will realize, wait, this essential process is not occurring. We can't have a cell which isn't segregating its chromosomes normally give rise to the next generation. If this cell is screwing up, if this cell is abnormal, it should not, be providing genes for your offspring. It should fail. And so there are error correction mechanisms, and this is part of it. Okay, so in the second stage of prophase 1, that zygatine, now you see the pairing of these homologous sister chromatids. And I'm calling them homologous because they have the same banding patterns, they have the same genes, all right? And so, if the genes for hemoglobin or keratin or whatever genes are on this chromosome, the homologous chromosome that you got from the opposite parent. So your mother's chromosome 1 has the same genes as your father's chromosome 1. They can have different forms, like, for blood type O as opposed to blood type A, but they both have a gene for blood type. Okay. So here's, say, a pair of sister chromatids you got from your mother, let's say your father. Here's a pair of sister chromatids you got from your mother. Now, I hate this term, but for right now, we would call this one single chromosome. The minute they split, we call them 2 separate chromosomes. But as long as this pair is together, this is called one chromosome. So a pair of sister chromatids is the same as one chromosome. This pair of sister chromatids is called a chromosome. But now that they're stuck together, we use the term bivalent or tetrad. So a synaptonemal complex which will form, which will join this blue one to this pink one. This pink one is not joined to that blue one, right? So a synaptonemal complex will cause them to join, forming a bivalent or a tetrad. Now, something that's critical here, there's something that is essential, which was happening, known as independent assortment. All right. At the end of this, I will ask the question, why do so many organisms reproduce sexually? Why is there sex? And the answer, will be that this increases the variation in the offspring. So that is the reason for sex. If sex did not increase the variation in offspring, there would not be sex on Earth. There would not be males on Earth. Every individual would be female. Yeah, I mean, obviously not bacteria, but you get the idea. So something here has to provide variation. That's why sex is here. And there are two processes which occur in prophase 1. Once again, prophase 1 is key. Two processes in prophase 1, which then provide that variation. That is the reason for sex. Sex would not exist if it weren't for two processes occurring in prophase 1. When those homologous chromosomes form tetrads, they could line up like this. So you could get, so when you make gametes, you could have in one cell your father's versions of all the sister chromatids lining up on one side of the metaphase plane, and your mother's chromosomes lining up on the other side. You could do that, but you don't have to, because your mother's chromosomes don't hang out in a pink gang. Your father's chromosomes don't hang out in a blue gang where they all say, oh, all right, we've all decided we're going right. The pink chromosomes from the other are going left. Okay. Now, by dumb luck, if all those, here are just 3 chromosomes, obviously humans have 23. So if I were to represent this fully, it would be a bit more complicated. In humans, if these 3 all lined up together, that means, let's say that this is producing sperm, and I'll tend to use sperm more than ova because I'll see at the end, the ova are more complex. We have to deal with polar bodies and the like. So let's just look at the male for right now. So then some sperm would be produced, which would have only the father's genes, which are on these 3 chromosomes. And then some sperm would be produced, which would only have the mother's genes. Now they all have the same genes. So they all would have, say, like a blood typing gene, or a hair color gene, or a can you roll your tongue gene. But let's say the father has blood type O, black hair, and can roll his tongue. Let's say the mother has blood type A, has red hair, and can't roll her tongue. Well, then when sperm are going to then provide genetic information to the next generation, it then matters which sperm are you getting. Are you getting this one, giving information for blood type O, black hair and rolling the tongue, or blood type A, red hair and not rolling the tongue? And so the combinations of genes, is what's going to provide variations in the offspring. But the chromosomes didn't have to line up like that. Once again, the pink chromosomes from your mother don't have to hang out in a gang. So when those synaptonemal complexes form, they could form like this. So notice that 2 pink chromosomes are going to, go down when these chromosomes separate, but 1 is going to go up. So that now means that you're not going to have children who have blood type O and black hair and can roll their tongue. There could be, blood type O and red hair. So this sperm is now mixing the genes that the offspring will inherit. They won't just get the genes from the, forms of the genes from the father or the forms from the genes of the mother. So the sperm which will result from this will have the father's versions of some chromosomes, the mother's versions of other chromosomes. This is what's called independent assortment of chromosomes. You may remember that one of Mendel's principles was the principle of independent assortment. We observe that traits can independently assort. They don't have to travel together. That's why you get that 9:3 to 3:1 ratio. Why do traits assort independently? Because the chromosomes that have the genes determining those traits are assorting independently. This pink chromosome can go this way, even if the other pink chromosomes went the other way. And so the independent assortment of the chromosomes in prophase 1, as these bivalents form, this gives rise to the independent assortment of traits, which gives some of the variety which results from sexual reproduction. One of the reasons that sexual reproduction exists. So that is significant. There's one other process which happens in the next phase, known as pachytene. So in the pachytene phase of meiosis, not only are the homologous chromosomes attached in a bivalent, but now you will actually cut blue genes from the father's chromosome and then stick it on the mother's chromosome, but then in return take pink genes from the mother's chromosome and attach it to the father's chromosome. This is known as crossing over. When this is over, we will end up with what are called mosaic chromosomes. This chromosome is pink. It only has the genes which originated from the mother's chromosome 1. This chromosome is blue. It only has the genes which originated from the father's chromosome 1. But these chromosomes have blue and pink genes. And so let's say that, I'm just making this up, that on the short arm of chromosome 1, there's a gene for hair color, and on the long arm of chromosome 1, there's a gene for eye color. And let's say your father has black hair and brown eyes, and your mother has red hair and green eyes. All right. So as long as they stayed intact, then the pigmentation of the children should be either black hair with brown eyes or red hair with green eyes. Okay. But if you can mix those up, you can now separate the two genes so that you could actually then have, red-haired children with brown eyes or black-haired children with green eyes, okay? And so this is another feature. Now, I'm going to talk about how this happens, but just big picture. The reason that sexual reproduction is with us is because of the variation that it creates in the offspring. Because of these two processes, crossing over, all right, where you cut some of your dad's genes and paste it on your mom's chromosome and vice versa, because of crossing over an independent assortment, each pair of people, so you and someone else, a significant other, has the genetic potential to make more genetically different human children than there have ever been human children. So every sperm, every ovum, not only has this unique combination of pink and blue chromosomes, but a lot of them are mosaic chromosomes with a little bit of pink and a little bit of blue over here, a little bit of pink and a little bit of blue over there. And there's such variety there, such an incredible variety that you and someone else will make, together you could make more genetically different children than there have ever been human children. So this is an incredible potential for variety. Once again, variety is the reason for sexual reproduction. So prophase 1 has these two very important processes, the independent assortment of chromosomes and this crossing over, which we see here in the pachytene phase. All right. Now, this is not an accident. We make this happen. All right. So there are going to be enzymes which cut and paste, so that we have, bridges happening here. And you end up with some paternal DNA on a maternal chromosome, maternal DNA on a paternal chromosome, etc. All right. Now, not only does it happen, it actually has to happen. If this doesn't happen, you won't make sex cells from this meiosis will fail. So this cell, if it's going to try to make gametes, unless this is happening, it won't. Now, one other, I said the reason for sexual reproduction was that, was the variety. Just mechanically, there might be one other aspect, which is why this originated in the first place. This is the way that you make sure that you've got the right numbers of chromosomes. So like if you want the right numbers of chromosomes in a gamete. So just one chromosome 1, one chromosome 2, one chromosome 3, not 2 copies of chromosome 1 and 0 chromosome 2s. You could still get 23 chromosomes, but just have the wrong ones. Here, if you're going to line them up and say crisscross the same genes, if they don't match, all right, so if we're matching up chromosomes that don't pair well, you won't be able to do this, and thus it'll fail. So it's also a way that you can make sure that you've actually got chromosome 1s pairing with chromosome 1s, because you couldn't cut and paste if it were anywhere else. So we actually make chromosome breaks, all right? Because once again, you want to make sure that when you're forming a tetrad, all right, that you're not forming a tetrad like this, all right? So this is chromosome 7, this is chromosome 1. You don't want them to pair, all right? You want the chromosome 1s to pair and the chromosome 7s to pair. So we need a way of making sure that the gametes have the right chromosomes, not just random assortments of 23. Otherwise, they would have too many hair color genes and 0 blood typing genes. Now, there's a type of mutation repair, which we can discuss later, that was actually invented in bacteria. So it's kind of funny that this is essential for sexual reproduction. But this essential piece of sexual reproduction is actually modifying something which is just a mechanism for mutation repair that evolved in animals that don't, or evolved in bacteria that don't reproduce sexually. All right. So what you're about to see is a type of mutation repair in bacteria. We not only use it for mutation repair, we also then use it for this crossing over. So if you want to repair your DNA, you say, oh, well, where's a good copy that I can use as a model? Well, you say, oh, okay, oh, here's a double-stranded which hasn't been damaged. So look, they match. So I'm going to drag matching the damaged section to a matching good section, and I'm going to then cut the good, one of the 2 good sections, because that one's double-stranded, it has 2 strands. I'm going to cut one of the 2 double-stranded and paste it on the damaged area using a protein called Rec-A. Rec-A is used in mutation repair, but once again, it is now also then used for the crossing over, where when we line up the chromosome 1s, Rec-A will then cut part of the pink chromosome pasted on the blue, and vice versa, so that now we have those mosaic chromosomes. Now, so like I said, we introduce breaks. We actually cut our DNA and make breaks, and we do it on purpose so that we can then see if these sequences line up. If they don't line up, then you've paired the wrong chromosomes, and this should not be a sex cell which gives rise to the next generation, because obviously it's messing up, all right? And we don't want that to now, lead to, genes combinations which are inappropriate in the next generation. So we actually require double-stranded breaks and for this synapse and recombination. So you cut your DNA on purpose. Not all of these cuts will end up being places where we do crossing over, okay? So we actually made some holes now that need to be fixed. And so that's why we're going to now incorporate this mutation repair modification, okay? So lots of double-stranded breaks per pair of homologous chromosomes, 160 in yeast, 400 in mice. So you do that a lot, all right? So if you have 2 chromosomes lined up when you're making sperm or making ova, you then make holes, all right? These are occurring at pre-programmed spots, all right? So other cells making sperm or ova will also have these happening, but not everyone will end up in a cross, a bridge and crossing over. So that will vary from, when this cell making sperm or ovary does it, it will form crossovers at different spots. So there's going to be multiple double-stranded breaks, but once again, some of them will be repaired without crossing over. So look, notice crossing over is occurring there, but not there. It could have, and maybe in the next cell that's making sperm, it will there. But that way, and note that the crossover events at 3 different sites can produce 3 different sets of mosaic chromosomes. So if all of the cells in, say, 3 different spermatocytes, so let's say these are primary spermatocytes undergoing spermatogenesis, all of them will develop a break point here, here, and here. But maybe this one only does a cross bridge here, which makes these chromosomes. And notice where the pinks and the blues are. If this one does it here, notice where the pinks and the blues are. If this one does it here, notice where the pinks and the blues are. And so thus there will be different mosaic chromosomes resulting in 3 different spermatocytes, which are beginning meiosis. So once again, this could happen 400 times in the chromosomes of a mouse. Most of the time, it would not result in crossing over. Some of the time it would have to. But that allows one spermatocyte to form crossovers differently from the next spermatocyte if it's a smaller fraction of them which are actually forming crossovers. Now remember I said that this is a mutation repair mechanism where you would then say, all right, let me fix this hole by dragging some of the good DNA from the neighboring strand in to repair that. But now in the process, I'm bringing blue DNA to fix the hole in the pink chromosome. And so that's where I will get that mosaic chromosome forming. So there are a number of genes involved in this. And once again, you physically make holes in your DNA and physically drag some of one parent's DNA to the chromosome of the other to fix this. Now you can repair it without crossing over, just drag in a little tiny section to fix the hole, or once you fix the hole, you kind of just keep going, all right? And so once again, you have a hole and you're gonna fix it, great. Most of the time when you fix it, it doesn't end up dragging, like a whole nother section of the chromosome with it. But that can then happen, and that's what we're seeing here. So instead of just fixing the hole, now we have a crossover event occurring where now it won't just be the hole that's fixed like a little blue piece here. It's now going to be a much longer blue piece which ends up being displaced from there. These cross bridges are known as chiasmata, and at least one has to form in a pair of sister chromatids, or else this cell will fail and not produce gametes. So this is not an accidental thing. This is how your cells are making sure that they've correctly paired, because if you're saying, I need to fill a hole with a matching section, if it's not a matching section, you may say, oh, I have, I think, matched chromosome 1 with an inappropriate chromosome, and so therefore this can't continue. Okay. So the Synaptonemal Complex, if you recall, started in leptotene, the first phase. It was completed in pachytene, but zygatine when was a lot of it was occurring, and then finally in the 4th phase, diplotene, it will dissociate. So that the crossing over has been resolved. And so now that these can potentially separate once we get into metaphase. So in diploatine, the synaptonemal complex is breaking down, although those chiasmata are still in place. Now, something that can happen is, and this happens in women, women started making ova before they were born. So in a woman's ovaries, before she was born, the ovaries actually began meiosis. So they went through leptotine, they went through zygatine, they went through pachytine, and now here they are in diploatine, and they could then pause there. And so I'll get to this again later. When you compare the making of spermatogenesis to the making of ova, these cells from an ovary, they are paused in meiosis. Meiosis started, and then around birth, paused, and will remain paused until menstrual cycles, a decade and a half later, decades later, if this is, oocytes which a woman will produce later in her life. So meiosis can pause. And so in this phase, diploatine, some cells can undergo meiotic arrest, which obviously, talking about other animals, obviously there might be like a mating season, and then you might be an amphibian waiting for spring rains, or et cetera. So there might be like some triggers. So you want to be ready, to produce, gametes or tie it to a certain, time. And so here you can start the process and then pause it. And then in women, then over the course of a year, then these follicles can then become reactivated, and then later then be activated by hormones to complete menstruation. But I'll get to that at the end. At diakinesis, then, the chromosomes will shorten further as the nuclear membrane breaks down, and is similar to the prometaphase of mitosis. So thus, prophase 1 is so significant that it's broken into these 5 phases. And by the end, then microtubules are attaching, so that the tetrads can now move along the to the metaphase plate. Now, this is then different between mitosis in the prophase of mitosis and prophase 1 of meiosis. Once again, in prophase 1 of meiosis, this is where we formed so here are the similarities. So the chromatin is forming chromosomes, the nuclear membrane is breaking down, the spindle fiber is forming, so that that is constant. But then there are then those differences, where in prophase 1, this is where the homologous tetrads are forming. This is where the synaptonemal complex are forming. This is where crossing over occurs with chiasmata. That does not happen in prophase 1 of mitosis, nor would it happen in prophase 2 of meiosis. So once again, these are 2 things which generate variation: the independent assortment of chromosomes and the crossing over, which are major reasons for sexual reproduction. They're happening in prophase 1. Now, in metaphase, the chromosomes line along the middle of the cell. In prophase, in mitosis, they line up like this, where the chromosome 1s, the pair of sister chromatids of chromosome 1 you got from your father, they don't go anywhere near necessarily the chromosome 1s you got from your mother. They are independent. Whereas, remember, in meiosis, they are forming a tetrad. So here we have the metaphase plate in metaphase 1, the first round of meiotic division. The sister chromatids are still paired to their homologs. So notice that there is a difference between mitosis, where they don't have to be anywhere near each other. between metaphase 1, or metaphase of meiosis 1, the first round of cell division. By the time we get to the second round of cell division, we no longer have tetrads anymore. And so this formation is only happening in metaphase 1, not in metaphase 2. Metaphase 2 just has, the single pair of chromatids for chromosome 1 lining up along the metaphase plate. Right. And so the tetrad still exists in metaphase 1, but then in anaphase 1, the tetrad then disassociates, so that now one pair of sister chromatids is going into one cell, and another pair of sister chromatids is going in another cell. All right. So once again, in mitosis, all right, the tetrads are not forming. In meiosis, we have those tetrads because of the synaptonemal complexes, and then the chiasmata forming. But then in as metaphase becomes anaphase, then those tetrads will separate. And once again, this is giving them that variation in the offspring. So notice, as a result, some of the daughter cells will now only have pink versions of, say, chromosome 1, as opposed to while other cells making gametes will only have, say, blue chromosomes. So here we are, anaphase 1, and now the big difference with anaphase and mitosis. Anaphase and mitosis, the centromeres, they dissociate, and the cohesins are cut, separating the sister chromatids. But notice that in meiosis, the first anaphase will keep the sister chromatids stuck to each other. It is the tetrads which dissociate. So the one pair from each tetrad will go up, one pair from each tetrad will go down, but the sister chromatids will still be attached to the centromere. So that is a difference between anaphase of mitosis and anaphase 1 of meiosis. So if we were calling a pair of sister chromatids a chromosome, this is then called the reduction division, because now this is now just counted as one chromosome, and this is just counted as one chromosome. So now the daughter cell, instead of having two chromosome 1s, it just has one, because we're counting the pair of sister chromatids as a single chromosome. So the centromeres are still attaching the sister chromatids. The tetrads will separate as the pairs of sister chromatids are moving to now the opposite sides of the cell. So that's anaphase in meiosis 1. In anaphase and meiosis 2, that's when the centromeres separate, the cohesins are cut, and now you have no longer a pair of sister chromatids. They are now recognized as individual chromosomes going to opposite poles of the cell. So anaphase of meiosis 2 is similar to anaphase of mitosis, where once again the sister chromatids are separated and pulled to opposite sides of the cell. And so here I compare them. The separation of the tetrad is what occurs in anaphase 1. So meiosis 1 has some unique features here. So in telophase, now we have separated chromosomes that we then can simply wrap in nuclear membranes and form a new nucleus. In meiosis, the first telophase will now enclose these chromosomes, but once again, now it is said to be a reduction division, in that there will just be one of these chromosomes here, one of these chromosomes here. These are not considered separate chromosomes yet, because the cohesins have not been cut. There is no S phase in the split between meiosis 1 and meiosis 2. In meiosis 1, one cell divides into 2, and then it will then start another round of cell division, but there is no S phase, so there are no additional sister chromatids made. So in telophase of mitosis, we now have all of these chromosomes, and they're separate because at anaphase the cohesins were cut. But because in Anaphase 2, the cohesins haven't been cut, once one cell divides into two, that's the first round of meiosis, these sister chromatids are still combined. It is then the second round of cell division where the cohesins are cut. And so now we just have one of the sister chromatids, now a sister chromosome here. And so now we have 4 daughter cells. There was no duplication of the DNA in the S phase between meiosis 1 and meiosis 2, or else you would have more DNA here, more chromosomes, and obviously you don't. So I have a couple of videos, as you just saw, which compare those, because I think, you could get confused. There's prophase, metaphase, and anaphase, and telophase in mitosis, but then also in meiosis 1 and meiosis 2. And therefore, then here are some comparisons of those. Now, while it would be appropriate for me to now, say, talk about how meiosis occurs in, say, men and women, I also want to hit on a couple other topics and make all of them short, so I'd like to start wrapping this up. I do teach anatomy and physiology too, and so in one of the final chapters on the reproductive system, obviously the male reproductive system and the female reproductive system can be covered in much greater detail, and I do have videos on that. But once again, I think the appropriate level for this class is to go through it much more quickly than what other videos do. But obviously, if you'd like a more in-depth look at them, I have, once again, videos on that. So in males, there are stem cells called spermatogonia. They continually replenish themselves in males, which is why men can make hundreds of millions of sex cells per day for their reproductive lives. The factor which limits the number of sperm man makes in his life is how long he lives. The longer he lives, the more sperm he makes. The same is not true in women. The spermatocytes, the spermatogonia, those stem cells which replenish themselves, they then make this cell called a spermatocyte. This primary spermatocyte starts meiosis 1. It goes through prophase 1, and in prophase 1, the sister chromatids line up. And once again, they could be independent assortment. Crossing over can occur. So then we go through, this is now metaphase 1. We go into anaphase 1. Notice that in anaphase 1, the sister chromatids haven't been separated from each other yet as they would have been in mitosis. And so here we have the cells divide then in telophase 1. And then we now have secondary spermatocytes. So one primary spermatocyte cell divides into secondary spermatocytes. And once again, the independent assortment and crossing over occurs in prophase 1. Now there's no replication of the DNA in the S phase here. So in interkinesis, the split between when the first cell division occurs and the second cell division occurs, no more chromosomes. And so then now prophase 2 goes into metaphase 2, all right, where we line up in the metaphase plate. Notice there's no tetrads anymore. That was in meiosis 1 in the primary spermatocytes, not in meiosis 2 in the secondary spermatocytes. And so then when anaphase 2 comes, that's when the cohesins are cut, and then these sister chromatids are now individual chromosomes, and then they are then separated. Now notice the difference. All of these spermatids have a long chromosome, chromosome 1, a short chromosome, chromosome 7, but the cell that started off had 2. The cells that started off had a blue one and a pink one. It was diploid, whereas this cell has a blue one or a pink one or a mosaic, but it only has one big chromosome, one short chromosome. So this is now a haploid cell. So in testes, meiosis is called spermatogenesis. And the primary spermatocytes divide once, doing meiosis one to make 2 secondary spermatocytes. The secondary spermatocytes then divide to then make these haploid spermatids. Now, spermatids are haploid. That's good. That's the goal, but they can't swim. And so in testes, then a second process occurs, what's known as spermiogenesis, where the shape of these spermatids is modified, creating haploid cells with a flagellar tail, a midpiece, some enzyme in the tip. And so the functional male gametes, spermatozoa, result after a second process which modifies the shape of these cells. The situation is far more complex in women, which is why, once again, in the reproductive system videos that I've made, I have a lot more material. So the oogonia all become oocytes before a woman is born. So while men have stem cells which replenish themselves, women don't. And so that's why at menopause, the ovary may essentially have used all of its potential gamete making cells and no longer be capable of making more gametes. Now, these oocytes, so this is a primary oocyte, that will then begin prophase 1, but then it will pause. All right, remember that it undergoes meiotic arrest, and it will not then pick up until later. All right, so prophase 1 starts before a woman is born. A woman starts making her gametes before she's born, but then it undergoes through meiotic arrest. So part of prophase 1 begins, but then it gets paused. After puberty, then with menstrual cycle, cells are triggered. We used to think that this cell would then pick up from its pause with each menstrual cycle. We now know that it's much more involved, that the cell kind of reactivates and may then develop for over the course of a year before then it starts responding to hormones of a menstrual cycle and then do the final stages in a month or 2 or 3. And so it's a bit more complex. Once again, I go through it later, but if this is the time period in which this oocyte is going, it will then complete then prophase 1, then do metaphase 1, anaphase 1, and telophase 1, and so then become these 2 cells. Once again, in prophase 1, the independent assortment of chromosomes and crossing over occurs. Once again, in anaphase 1, the sister chromatids are still tied together with cohesins. It was the tetrads which split, not the sister chromatids. Another difference, however, is that while meiosis in males produces 4 functional gametes for each cell that begins meiosis, that's not what happens in oogenesis, the making of ova. Because when the primary oocyte splits to make a secondary oocyte, one of the 2 daughter cells gets all the good stuff. One cell gets essentially nothing. So this is a daughter cell, just like this one, but they're not equal daughter cells. This one did not get much cytoplasm. This is a polar body, not a secondary oocyte. And although it may divide again, this will not produce sex cells, only this one. So there's no S phase where DNA is replicated in interkinesis, and then the meiosis 2 begins, prophase 2, metaphase 2. In anaphase 2, the sister chromatids finally separate as those cohesins are separated, and then we get telophase 2. Once again, this second round of division was not equal. So these 2 are sisters, but one of the sister cells got all of the cytoplasm and one cell got essentially nothing. So this is also a polar body. This first polar body may or may not have divided. So meiosis in females is different from that in males in many reasons. In one way, that instead of having one cell that begins meiosis produce 4 sex cells, one cell that begins meiosis produces 1 sex cell, the ovum, and then there are 2 or 3 of these small polar bodies which occurs. Now, another difference in females is the timing, because very often I think a lot of people feel that ovulation is the release of an ovum, and a woman feels that, say, during a menstrual cycle, she releases an ovum. That's not exactly correct. It's close to correct, but not entirely correct. This is what leaves the ovary at ovulation. All right. So it's the cell that's undergoing meiosis plus some epithelial cells from the ovary as well. Once again, another difference. In females, the meiosis occurs as the oocyte forms part of a larger structure known as a follicle using some ovarian cells. The sperm just travel by themselves. So sperm are their own separate units. Oocytes are maturing as part of a larger structure, the follicle, and even now still retain some of those follicular cells. But look at these chromosomes. They are still in metaphase 2. See that? That's metaphase. The chromosomes haven't separated yet. The sister chromatids are still tied together with cohesins. They're still at the metaphase plate. So this is not an ovum. This is not a sex cell. It's close, but not quite. The trigger for meiosis to finish will be the penetration of the sperm. Only when the sperm penetrates will then metaphase 2 go to anaphase 2, which goes to telophase 2, which then allows this cell, the secondary oocyte, to divide one more time. So one cell division has occurred. The first cell divided to make a secondary oocyte that got all the good stuff, and this little polar body. See that? These are sisters, but one cell got all the good stuff, one cell got nothing, and that will not become a sex cell. Once the sperm penetrates it, then this cell will divide again, but it will also not divide equally. So as these chromosomes separate, one set of chromosomes will stay with most of the cytoplasm, and that will then become the ovum. Another set of chromosomes gets pulled to make another polar body, which will not function in reproduction. So note that meiosis in females produces this ovum. This is a sex cell with 23 chromosomes, but then it also then provides 2 or 3 of these polar bodies, which will not be fertilized and not result in sexual reproduction. So here you have a sperm with 23 chromosomes, an ovum with 23 chromosomes, and then when these chromosomes are added together and form a nucleus together, it is a diploid nucleus with 46 chromosomes, 2 sets of 23. That is a zygote. That is the beginning of a new individual that will now begin by mitosis. So there are 46 chromosomes there, and then mitosis will cause that to divide. Now, once again, I have videos that go through this in much greater detail, but once again, this is far more complex in females because there are no stem cells which allow the oocytes to replenish themselves. The timing is different. Meiosis begins before birth, pauses, picks up during a menstrual cycle, pauses, and isn't finished until fertilization. The sperm penetrating, that was the trigger for the final step for the secondary oocyte to become the ovum. A woman who has never conceived has never made an ovum, a female gamete. She produces secondary oocytes at ovulation, but that's not quite a gamete yet. It still has to divide one more time. Also notice that the meiosis is occurring in association with other ovarian cells as part of a larger structure known as a follicle. All right, and notice that this is occurring at different times. So this oocyte is maturing while these have not. They will now mature in other menstrual cycles. Now, some animals, like say fish, they might release all of their eggs at once, whereas we humans, we don't. And so obviously these would not all mature together. So the cell which is released from the ovary at ovulation is not an ovum yet. It is close to an ovum. And so once again, I teach the reproductive system in anatomy and physiology too. And so I have lots of videos if you were interested, how is this occurring in the males? It is here in the testes, in the seminiferous tubules. That is where meiosis is occurring, that's spermatogenesis. And you can actually observe the cells as the spermatogonia become the spermatocytes that undergo meiosis. Then you have the haploid spermatids and then the spermatozoa with their elongated heads. So the process of spermatogenesis is occurring as the cells accumulate towards the lumen. Okay, once again, a lot more detail on that, but in other videos. Two other things I would like to just address very, very quickly. One is, why does sex exist? There are clearly disadvantages to having sex. So here, the trials of sexual reproduction, like going far away from your safe pond to lay your eggs, that's something that only this female snapping turtle is doing. The male is still safe in his pond. So the female is bearing the burden of reproduction. In mammals, the young develop inside the body of the mother, and there's lactation afterwards. So these are periods of decreased mobility for the female. That's a great energy expense. And so this potentially makes life harder for the female. In a world where it's hard enough to get enough nutrients for you to get through. So these are burdens that the females bear. Males bear burdens in sexual reproduction as well. For two individuals to find each other, very often one has to make a spectacle of themselves. So males are often brightly colored. They sing courtship songs. They have courtship behaviors, but this exposes them to predators. So males are very likely to end up dead as they attempt to reproduce sexually. Plus, then there are just advantages to reproducing asexually. You can be by yourself and still reproduce, because some animals end up on an island or on the wrong side of a mountain range or a river from the rest of their population. You can be by yourself and still reproduce. If you have good genes, you can pass them down to your offspring. You don't have to worry about, a significant other that might contribute mutations. And so there are lots of advantages to not having sex. The population grows more quickly. You can reproduce, by yourself. And there are disadvantages to both males and females in reproducing sexually. But it is that variation that then gives the advantage to sexual reproduction, because the world is going to change. All right, I'm in Northeast the United States right now, and as I look out my window, I think that it was only, say, 12,000 years ago where this was a sheet of ice. All right, so there was a sheet of ice, a third of a mile or more thick, that went from here to the North Pole. So in very recent times, this area has changed. The climate is now changing, and it's getting hotter than it has been in recent history. There are new diseases which get spread, from coronaviruses to AIDS to Lyme's to Ebola to Lassa to Zika to so the world changes. It gets hot, it gets cold. There's droughts, there's more rain, there's new diseases, there's new predators. The problem with asexual reproduction is that it produces clones. So mitosis is good at producing cells which were just like the mother cell, a clone, whereas sexual reproduction produces variety. Now, the reason that this is important is because the world will change. If all of these fish are clones of each other, then if there's ever an environmental change which affects one of them badly, it affects all of them badly, and the population fails. But thanks to sexual reproduction, that they vary. And so if the temperature changes, if the pH of the water changes, or the amount of oxygen, or a predator or a parasite, because they vary, there's the likelihood that some of them will survive and that the species will continue. So sexual reproduction is not here for the individual, it's here for the group. The group is likely to survive environmental changes, new predators, temperature changes, because of the variation. And so thus that variation in meiosis 1 is critical. Once again, you and a significant other could have more genetically different human children than there have ever been human children. That is an incredible amount of variety. So last point then is it was one-celled organisms which invented sex. So bacteria don't have sex. They are asexual. Okay, and sex isn't required. There are actually some animals that don't have sex. So there are some fish that don't have sex. There are some snakes, some frogs, some lizards which don't have sex. And this is a euglena, a protist, which doesn't have sex. Dinoflagellates don't have sex. So it is possible to reproduce without sex, but there are those advantages then to having sex. And that is why it evolved in the earliest protists. And so one-celled organisms undergo meiosis. And so the things that I've gone through here, the homologous chromosomes, synapsing, the crossing over, the 2 rounds of cell division, where now the daughter cells are now haploid with half the number of chromosomes, that evolved in protists. So here's a paramecium. They can evolve asexually by fission, but then they can undergo conjugation as well and exchange haploid nuclei. And so these one-celled organisms, without sex organs and sex courtship and sex ducts and secretions, etc., they're just making haploid nuclei, which can now exchange and then fuse to form diploid nuclei. So it was the eukaryotes which invented sexual reproduction. Now, there are certainly variations since then. So for example, if you took General Biology 2, I'm sure one of the things that confused you is that not only do plants have meiosis and they have, diploid cells and haploid cells, but the haploid cells don't have to be limited to sex cells. They can actually grow up. So when you look at a moss, the moss is actually mostly haploid. The green part is all haploid. It's like, if a gamete grew up into an individual, all right? Now, on these haploid strands, that's where sperm are made, where ova are made. They fuse to then make diploid structures, which now have double the number of chromosomes. So the green part is haploid. This is diploid. That then forms a stalk with a spore case. That's where meiosis occurs to produce haploid spores, and then the process occurs again. When we look at fungi, the same thing, that fungi can be primarily haploid. So haploid spores can then produce these haploid hyphae, which grow out into the material before you then get cells which have 2 sets of chromosomes, which then undergo meiosis to make haploid spores, and the process can occur again. So as we look at other organisms, there are variations. One of their variations is haploid cells don't have to just be limited to the sex cells. And so the reproductive cycles of other groups, can have variations. Now, I discussed this with sex linkage. It's kind of related here, so I just want to mention it in passing. Meiosis then evolved in the first eukaryotes. Plants do meiosis, fungi do meiosis, and animals do meiosis. Now, as you saw, there can be variations, like how long the haploid generation lives, and then even in animals there are variations, like what determines gender. Well, in reptiles, for example, it's very commonly the temperature of the eggs. So if you have warm eggs and cooler eggs, that could then make the difference between, say, the male offspring and the female offspring. There don't have to be sex chromosomes. That gender can be determined by, say, whether an egg is fertilized or not, say in bees, or a cytoplasmic factor. If there are sex chromosomes, while in humans and flies, the males are the ones with 2 different chromosomes. They're XY, and therefore make 2 different kinds of sperm. They are the heterogametic gender, and while females are the homogametic gender. It doesn't have to be that way. Now, before I get to a difference there, flies are a little bit different because even though the males are XY and the females are XX, the X and Y chromosomes in flies are not related to the X and Y chromosomes in mammals. And actually what's determining the gender in flies is the ratio of X chromosomes to autosomes. So in mammals, having a Y chromosome will probably make you male, at least chromosomally male. But in flies, it's having two X chromosomes then is what makes you female. See, here's an individual that just has one X chromosome. In mammals, this individual would be female, but in here, this individual is male. In mammals, having a Y chromosome makes you male, but because there are two X's, that makes this individual female. So even though there's X and Y chromosomes, what actually is determining gender in flies is the ratio of the X chromosomes to the autosomes, which is a little bit different. And then finally, here there are sex chromosomes in, say, birds or butterflies, but there it is the females which have the two different chromosomes and are thus heterogametic, and the males which are homogametic. So once again, say a genetics student has gone through meiosis before in high school in maybe General Bio 1, and then this was building on that foundation and adding a bit more in terms of details.